Good morning, everyone, and thank you for joining us today for our first seminar of this series for this year, 2021-2022. My name is Hardev Sandhu. I'm associate professor in the agronomy department, and I work over here at Everglades Research and Education Center in Belgrade. And our first speaker for our seminar series is Dr. Wagner Vendrame. Dr. Vendrame is a professor and assistant department chair in the Environmental Horticulture Department in Gainesville. And he's responsible for teaching micropropagation of horticulture crops and orchard biology and culture. And his research program is mainly focused on production and conservation of ornamental plants using plant tissue culture and cryopreservation techniques. He also worked with NASA for 5 years on spaceflight studies on board of the Space Shuttle Atlantis, Endeavour, and Discovery, and the International Space Station National Laboratory. Dr. Vendrame has also worked with the biofuel species for the development of biodiesel and biojet fuel, and saw palmetto, a medicinal plant for the production of prostate health supplements. In recent years, Dr. Vendrame's research is focused on cryopreservation and large-scale in vitro propagation of plants using bioreactors, as well as micropropagation of several ornamental plants. Dr. Vendrame obtained his BS in Agricultural Sciences and MS in Plant Physiology and Biochemistry from the University of Sao Paulo, Brazil, and his PhD in Horticulture from the University of Georgia. Thank you very much, Dr. Vendrame, for presenting your research and sharing your experiences with us today. The floor is yours.
All right. Thank you, Hardev, for the introduction. Thanks, everyone, for being here. And it's great to see some familiar faces. And so the title of my presentation, Combining Tools for Innovative Research. I'm just going to show you a little bit of basically everything that I'm doing and some things that I have done. I think that my approach to research is that I like to do things that are innovative, things that challenge me, that put me outside the comfort zone. And and sometimes these ideas, they pay off. Sometimes it's just a risk, they don't work. But I like taking those risks. And so I'm going to summarize to you, some of the work that we have been doing. Starting, I heard I've mentioned that these are the courses that I teach: orchidology, also called Orchid Biology and Culture for the graduate session. That's fully online. And it's a very popular course. It's kind of fun to teach. And this year, I took over the course that Dr. Mike Kane used to teach in Gainesville, Micropropagation of Horticultural Crops. Mike Kane retires, so I came to Gainesville and I assumed that position and started teaching this course. And this is interesting because now I'm trying to introduce into this course also some elements of leadership that as I work with several growers, they always mention that our students come well prepared in terms of technical knowledge, but sometimes when they need to manage people and deal with people, that they have issues. They are not prepared for that. So I was part of the Roche Teaching Scholar Program, and I also participated in different leadership programs like Lead IFAS, Lead 21, and that helped me to learn a lot about leadership and incorporate those leadership concepts into a course. And it's been fun teaching now micropropagation. We're doing that kind of approach to leadership into teaching. So I start with micropropagation, which is something that I have worked for so many years. And so currently we have different projects going on. This is a project with an orchid, Brassavola nodosa. That's a species that is very beautiful and interesting. So this is a project with a PhD student. Actually, JJ, he used to be Dr. Zanalden's master's student. Then he came to work with me, and he's been doing some great work in micropropagating this orchid using both semi-solid and liquid culture, but also bioreactors and also cryopreservation. So he's about to finish his program, and hopefully he will have some really good results to present soon. We also work with palms. We have been working with a nursery in South Florida with a lipstick palm of Cyrtostaxis renda. As you can see, this is a beautiful palm. Everybody loves this palm, but it's very sensitive to cold. And this grower, during a cold spell, I think back in 2011, he lost a lot of palms in his nursery, but several palms survived. So he had this kind of belief that maybe these plants could have some cold tolerance. So we talk about maybe micropropagating that plant and evaluating for cold tolerance. So that's one of the projects that we have been working on. Another palm that we work with is Atalea crasispata. That's a palm native to Haiti, basically endangered. There are probably a few plants left in the country, but down at track, we had maybe 11 plants. And so we started developing also a system for micropropagation of this palm. Saw palmetto, as Hardev mentioned, that's a project of a graduate student. The saw palmetto seeds or the berries, the fruits, they are used for this saw palmetto prostate health supplement. It's very popular. The thing is, most of the by the way, this palm is native to Florida, so you find it everywhere under, pine lands and sand areas. Sometimes used as an ornamental, but mostly for extraction of the berries. And the problem is most of the seeds are extracted from the wild, from native habitats. There's no really commercial production for the production of the seeds, of the fruits. So we're developing a long-term project that is for development of commercial practices or best management practices for cultivation of this palm. Another project that's with ornamentals. We've been working with different nurseries on micropropagating variegated foliage species, such as cordyline as one of them. The other one is monstera. These plants, they fetch like good price at retail stores. For example, a monstera plant like this, is like you see in the picture, variegated, could sell for up like $500 sometimes. And I have seen on some websites them being sold by like $2,000, $3,000. So it's a very valuable plant, and we have been developing a system for micropropagation of this plant. We finally got a system going, and soon we're going to start looking at bioreactors. Clerodendron is another ornamental species that we're also working with, also in collaboration with a nursery in South Florida. And hemp. Of course, hemp is popular. Everybody's doing something with hemp today. But as this industrial hemp pilot project started, Dr. Zach Brin down in Homestead, he started working with hemp. And we started a collaboration, mostly working with in vitro germination of seeds, but also micropropagation. And we have advanced to a point now that we're getting really good rates of multiplication in vitro, and the plants are coming with really good quality. However, hemp is a crazy plant. That the behavior in vitro is insane. So you have some plants that grow really well coming from the same genotype. Other cultures will not grow that well. Some start flowering in vitro. So it's an interesting thing. Indigo this is something that is not really an ornamental, but this is a plant from which you extract the leaves, and from the leaves you extract a dye for dyeing clothes, like your blue jeans, for example. This was a partnership with a company called Stony Creek Colors. Their idea is to expand production of indigo for like sustainable production and extraction of a natural dye for clothes. And they wanted to test the plants in South Florida. So we did some trials to see if the plant will be really grow well. And it did, actually, with basically no pests, no diseases. And we proposed that as an alternative crop for vegetable growers who were desperate to have some alternatives to their vegetable crops. And the company, the last that I heard, they were in talk with growers and doing some commercial testing. So I believe that things should be going well. In terms of cryopreservation, first, what is cryopreservation? Is the storage of biological material at ultra low temperatures, which is minus 196 degrees in liquid nitrogen, or in the vapor phase, which is about minus 150. Cryopreservation is very safe, is cost efficient, and allows long-term storage of several types of plant material, like in vitro shoots, callus, somatic embryos, seeds, pollen, and it has several purposes for, plant breeding, propagation, etc., conservation. We have done most of our cryopreservation work with orchids and orchid hybrids and orchid seeds and protocorms and pollen, also some orchid species. And here is like just a brief list of some of the publications. We have some more that came out more recently. Also have some book chapters and review papers on the topic. So that has become like one of the main areas of research in our lab. But basically what we use is a system called cryopreservation by vitrification. In this picture, you can see here, this is a seed capsule, orchid capsule containing the seeds inside. The seed of orchids are very minuscule. They're very small. So in this capsule, you could have maybe hundreds of thousands of seeds, sometimes 100,000, 200,000. There are some orchid capsules could have numbers up to the millions. So we extract the seeds. Here you see the seeds are like a little powder. We test the viability of the seeds using tetrazolium. And here is interesting because you see the embryos, they are dyed with the color red, showing that they are viable, and you don't see anything around it because orchid seeds don't have endosperm. They rely on the association with mycorrhiza for germination in the wild. But when we put them in vitro, of course, we are providing all the nutrients necessary for the seeds to germinate. But for cryopreservation purpose, we not only do the viability, but we also do the germination test to see if they match, if the tetrazolium particularly is a good measure of viability. And then we go through the process of vitrification, which is a solution comprised of glycerol, ethylene glycol, DMSO, in MS medium with sucrose, and that allows some osmotic balance with the cells and prevent the formation of intracellular ice in the cells. And that allows that content in the cell to be super cool, but not frozen. And therefore, you can store material in liquid nitrogen for indefinitely, in theory. Of course, our evaluations, our experiments, they go from as low as 14 days to a couple of months or 3 months. And that way we can, bring those plants back and look at their germination and how they grow, how they develop. In this illustration here, you see these are protocorms of orchids. These are like round green structures that they start forming once the seed germinates. And you see the little leaflets forming and some, primary roots forming too. And again, we follow their development. We put them in the greenhouse and we evaluate. And we have been very successful with those protocols. More recently, also, we work on cryopreservation of rhizomes of bananas. And that's another project was done in collaboration with a visiting researcher from Brazil. And it was very successful. We here showing that we have effective treatments combining PVS2, which is a pre-vitrification solution, with another compound, fluoroglucinol, that enhances survival after cryopreservation. In this case, we have 100% survival, and the shoots and roots, they were really of good size and good health. Now, switching to bioreactors, this is something that we started back I would say 2016, maybe 17. This started actually with a postdoc who came into my lab, Dr. Elio Jimenez, who now works for Florida Crystals. And we started discussing the idea of using bioreactors for producing plants for micropropagation. And why is that? Well, bioreactors, instead of using agar, we use a liquid medium, which improves nutrient uptake. Also, the size of the vessels are large enough that improves aeration, therefore improving also multiplication rates and biomass production. And by having the large volume and also have separate vessels for a medium and your plants, you reduce plant manipulation, so you reduce contamination. And it's a system easy to scale up and resulting in reduced production cost on a per plant basis. So the way the system works is like this. We use the system from, it was developed by Cetis, that's a company from Belgium. So in this illustration, in the top here, you see what we call stationary phase. So there are two flasks. The bottom flask carries the liquid medium. The top vessel is where you put your explants. So during an immersion phase, you have compressed air coming into the medium vessel, and that compressed air pushes the medium to the top vessel. Therefore, irrigating your explants with medium. And you can control the frequency of immersions and the duration of each immersion. And everything is controlled by computer. So after the immersion phase, the system turns off and the medium returns by gravity, because I don't know if you can see, the flask is slightly slanted, so allows for the medium to return to the culture vessel by gravity. You can also do pulses of aeration by injecting air into the top vessel, and that again allows better aeration of your explants. And all that contributes to better growth and development. This is an illustration showing Phalaenopsis multiplication using the Cetis bioreactors. And as I mentioned, everything is controlled by computer. Here on the left side, you have like the platform that we use for controlling it. And at the bottom here, I know it's small, I don't know if you can see, but in this case, you have the running conditions for banana cultures. Here you have the frequency of immersion, in this case, every 240 minutes, that means every 6 hours, and the duration of each immersion, 60 seconds, that means one minute, right? And the ventilation or aeration every 480 minutes, every 8 hours, with a duration of 20 seconds. So again, it's nice, the system, because you can develop separate parameters for immersion for different cultures. In this case here on the side, you have banana, bromeliads, sugarcane. We also did orchids. And this whole thing with sugarcane led to Florida Crystals. They became interested in it, and we did a small test for them. They had two varieties that they really liked, but they were very susceptible to orange and brown rust. And we proposed the production of clean plant material using bioreactors. And they gave us a small grant to show them that we could produce like a couple thousand plants. And we ended up producing, I think, in the range of 4,000 plants very easily. And they got excited with this project, and they start funding a larger project. And Dr. Elio Jimenez was leading the project. And halfway through, basically Florida Crystals was convinced, and they decided to hire Dr. Jimenez, and he's now at Florida Crystals. And that's a shot of their bioreactor room, which I think they have about 500 units running nonstop, and they're producing the range of 2.5 million plants per year. So it's very impressive. And it's nice to see a project that started with us and led by Dr. Jimenez, that now it transferred to the industry. And that's a shot of our vessels. You can see the bottom here is the culture vessel. On the top here, you have sugarcane plants. That's another shot from the side. And what is interesting is that on the top here, we start with 80 explants, 80 small shoots. They're trimmed and we put them there. So in the period of about, I think, 8 months, we get this vessel filled with about close to 1,000 plants. So the rate of multiplications are really, really good. Now, switching to micropropagation, microgravity, and I'll tell you a little bit about the different experiments that I developed and how this whole thing started. Well, the first thing is that we wanted to look at the effects of microgravity in the structure and growth of cell cultures. So we wanted to look at changes in cell structure, changes in cell growth, and later on, we even started thinking about looking at differential gene expression. This whole idea started because I was contacted in 2007 by a retired researcher from the USDA, and in collaboration with NASA and Boeing, they wanted to look at stem cells, human stem cells actually, but the difficulties of working with human stem cells and all the, the difficulties that come with it, they decided to look for someone who was working with cell cultures. And for some reason, they came to me and we put together a small proof of concept experiment just to show how those cells could develop in microgravity. So our strategy was to do this preliminary study with suspension cultures. At the time, we didn't have a lot of time to prepare them. So we used cultures that we had already in our lab, like Encyclia plicata is an orchid, Caesalpinia pulcherrima is a dwarf poinciana, and Tabebuia aurea is one of these flowering trees that has really beautiful flowers. And Arabidopsis was provided by a researcher, Dr. Rob Furl, here in Gainesville, who was already doing some work also with NASA, but he was nice enough to provide some of those cultures. So with that in mind, we have those cell cultures going on, so that cut us some time and we had everything ready. And we collaborated with Bioserve, which is company out of the Department of Aerospace Engineering at the University of Colorado. They developed hardware for flying experiments, and that helped us a lot because they had all sort of different hardware that we could adapt for flights, so we didn't have to develop new or engineer new hardware. They also dealt with all the logistics pre and post flight, so all the documentation and permits that are necessary, they work with that, and that way it frees free time for us to just focus on the research. And this first experiment, it went to the International Space Station National Laboratory for a period of 3 months. We used this device called OptiCells, which is basically our 2 gas permeable polystyrene membranes. And you see the picture here. And they're attached to a standard frame, which is the size of a microtiter plate. And they have on top here this kind of resealing access ports. That's where we would insert our liquid cultures. And the way it was assembled, we had an array of 3 OptiCells together with these plates in between. They serve like as mirrors. And you see a cutout here showing the second plate. This is because attached to this set of OptiCells was a set of cameras that they could shoot a video real time while in space, and we could access those videos and monitor the growth and development of the cells. So it was kind of cool. It was kind of a high-tech thing and an interesting way of doing an experiment. That's showing the array, how it was set up in this little device called the CHAP. We will have 3 OptiCells on one side and another 3 on the other side. Some of these OptiCells will be just with plain culture medium, and the idea is that if we had medium depletion in any of the OptiCells, we could use some peristaltic pumps to inject medium into the other cells. So this is how they look when we were assembling them in the laminar flow hood. And that's them with the cover on. And again, here showed just a detail of the camera setting that was attached to this box, and we could collect video information. Everything was put in this CGBA, which is called, means commercial generic bioprocessing apparatus. When you work with NASA, everything has acronyms. They have like it looks like a Bible. It's a book of acronyms. They use acronyms for everything. After a while, you start getting used to some of those acronyms. But anyway, this apparatus allows the gas exchange. You have an air inlet, an air outlet, and also temperature control. So that helped us a lot. And because our cell cultures didn't need light, they were in the dark, so that was a perfect environment. So this just showing how these little boxes are arranged inside the CGBA. And also they had other experiments going on here. So they are able to arrange multiple experiments in just one box. So that was launched on August 8, 2007. It stayed at the International Space Station for 3 months and came back in November 2007. And that was a lucky shot when the shuttle had just landed and opened the parachute. And here at the bottom, you see the payload experiment truck. That's the truck that actually goes and retrieves all the experiments and brings them back to the lab. So that was exciting to be there and seeing the shuttle landing. And here is some of those preliminary results. Showing here on the left, these are the cell cultures that stay on the ground. And on the right, the cell cultures that went to space. Same cultures. This is the Caesalpinia pulcherrima, the dwarf poinciana. And what is interesting is that the cells that stay in the ground, they tend to spread around the opticell and grow in a more two-dimensional fashion. But when in space, they tend to cluster together in a more three-dimensional fashion. And they start forming what it looks like to us pro-embryogenic masses. So they kind of have some sort of differentiation. And that was interesting to see. The parallel that the guys that contact me for this, what they wanted to think about is that, let's say if we could culture human cells, stem cells, and on that kind of fashion. So imagine that you need a liver transplant, and we could just harvest a couple cells from your liver, send them to space, and using this kind of three-dimensional growth fashion, form a brand new liver that you could bring it back, and you wouldn't have any rejection issues with transplant. But again, this is like a stretch in what we were trying to do. But that led me to another part of the research, which was working with Jatropha. And we sent some Jatropha cell cultures into space. But everything started because Jatropha is this shrub or a small tree. It depends on how you see it. The tree can grow as tall as an avocado tree. But it has really good properties, particularly the oil content in the seeds has been proved to be excellent for biojet fuel and biodiesel. But the plant, basically any part of the plant has some interesting properties. It has also some medicinal properties. There are some compounds in the plant that have shown to have some anti-cancer properties. Boeing did a study using oil from Jatropha. And here you see on top, they look at the properties for Jet A and Jet A1, which is jet fuel, pure jet fuel. And the properties include freeze point, thermal stability, viscosity, contaminants, and heat combustion. And they compare to Jatropha alone, a mix of Jatropha with algae, and Jatropha, algae, and camelina. And this was actually done on flights, Air New Zealand. I think this was Continental Airlines, Japan Airlines. So in summary, for all the properties evaluated, the Jatropha oil either meets or surpasses the standards for aviation. And that, again, that's why Boeing and NASA were very interested in Jatropha. We started the study back in 2009. We planted about 111 sessions of Jatropha at Trek. And you probably know the soil in Trek is basically rock. But we put the plants there in June 2009. By November, they were already producing some fruits. So it was really great to see how the plant grows really well. Is a good plant. We also look at oil content. This is just a summary of different accessions. We had 111 accessions from Asia, Africa, Central America, and South America. And looking at oil content, we identified a few like the Mexican and the Mozambique accessions that had slightly higher oil content in the seeds. In 2011, these trees were already much bigger. We did a lot of assessments looking at reproductive characteristics, production of male flowers, female flowers. We also performed some crosses and generated some hybrids. And that was when the whole thing about biofuels was going on. But then all of a sudden, of course, oil prices dropped and our funding also disappeared. So we never completed. But anyway, the idea with Jatropha is that it is still considered a wild species. So some issues like cold tolerance, flower synchronization, fruit maturation need to be addressed towards development of commercial cultivars. So a lot of breeding and genetic improvement is still needed for Jatropha. But our question at the time is that could microgravity assist in accelerating genetic improvement in Jatropha? So our hypothesis that microgravity affects the gene expression in vitro cultures of Jatropha, and the differential gene expression in vitro Jatropha cultures under microgravity have implications for genetic improvement of the species towards domestication, adaptation of the species to abiotic stresses on Earth after return from space flight, and I'll explain that, or adaptation of the species to the space environment, the microgravity, thinking about long-term missions such as Mars. And everything here has to do with basically differential gene expression. So our first two flights with Jatropha cultures, again, they were a proof of concept. We had a lot of testing with different hardware. We were using these tubes here called fluid processing apparatus that were housed in these gaps called group activation packs. These tubes allow us to have cell cultures, and another part of the tube will have a fixative. So while in orbit, astronauts could attach a crank here and give a few turns, and then this piston would move and inject the fixative into the cells. So we had two flights that evaluated that. One was February 2010, STS-130 with Endeavour, and May 2010, STS-132 with Atlantis. We also look at effects of microgravity on cell growth and development. We had about 72 samples, 68 days in orbit. So we look at cell growth and development. We started with some leaf explants. We look at how the callus production was in the cells. And what happened in one of those flights is that it was supposed to stay there for just a couple of months, but the shuttle that was bringing the cultures back got delayed because of some technical issues. So these cultures ended up staying in orbit for about 312 days. And by that time, I thought that my cultures would be dead when they returned. Surprisingly, they were not dead. And actually, they start differentiating in orbit and producing shoots and some structures that to us look like somatic embryos. So that was an interesting observation. We were able to regenerate some of those plants. And then we evaluated much better our hardware. We still use those gaps called the group activation packs, but we adapted them for Petri dishes. And with that, we were able to fit more samples and do better evaluations. We ended up publishing this study in 2013 in the journal Advances in Space Research. And then led us to the last two flights, which was STS-133 with Discovery and STS-135 with Atlantis. Atlantis was the very last shuttle of the shuttle mission, the space mission of NASA. So while the 133 stayed 111 days at the International Space Station National Lab, The one that went with Atlantis stayed with Atlantis at the mid-deck, and only for 14 days, which was the duration of the flight. We had 56 samples in 133 and 72 in 135. And we did several comparisons, comparing ground versus space, effects of culture media, and even the length of exposure to microgravity, comparing 14 days with 111 days. We also were able that some of the cultures that came back from this first flight, they look really good. So we separated some of them and sent back with Atlantis for a second flight. So we had also data that counts 111 days plus a few days back on Earth, and then another 14 days back in space. Prior to doing all this work, we need to do some sequencing because there was not much information available for Jatropha. So we perform a transcriptome sequencing with the help of the Roy J. Carver Biotech Center, University of Illinois, which was interesting. I wanted to do with ICBR, and they help us here, but they suggested that we did with Illinois because it would be much cheaper, especially because we had to do a lot of samples. But it was good. We got a lot of information. This became actually the dissertation for one of my graduate students, and we published this. And when we look at the results, this is the plot showing the ground signal versus the flight signal. And the red lines indicate the change in differential gene expression, we call differential gene expression, either at the twofold or fourfold. Then this is just to illustrate one of the comparisons that we did, but we did the gene ontology. We look at the biological processes, cellular components, molecular functions, and that generated tons of data, but too much data that honestly I was not able to evaluate everything. More recently, I joined the NASA Gene Lab working group, and in collaboration with Dr. Richard Barker of the Gilroy Life Science Lab at the University of Wisconsin, he was able to upload all the data into the TOAST, which is a database basically, and generated these nice graphs. And now we're starting to look at all the genes that were differentially expressed. But I'm talking to you about like tens of thousands of genes. And so it's a lot of data to be analyzed. But we're moving forward. Details of this experiment is in the NASA website. If you go to the Space Station Research Explorer on nasa.gov, and you do a search for Jatropha biofuels, you probably will find the information there. The link is there. And that's it. Thank you very much for your attention. Again, my goal here was just to show you how I started using all these different tools for developing new ideas, like from micropropagation going through bioreactors and then space biology and cryopreservation also, not only for seeds, but also for shoots and other applications. So with that in mind, I'll leave it now open for any questions.
Thank you very much, Dr. Vendrame. Any questions, please feel free to ask. Just unmute your microphone and you can ask your question.
Wagner, I had a question.
Uh-huh.
So you mentioned the soils in Homestead. What did it take to make this Jatropha successful? I mean, what kind of management strategies that you had to do?
Well, the first thing, if you recall the picture, you see that the holes were perfectly, drilled. We had to drill the holes. So we bought a tractor with a drill and drill the holes. We basically, we grew them in the greenhouse and make sure that the plants look really healthy before transplanting to the field. Once we put in the field, basically we didn't do anything except basic fertilization and irrigation when needed. You know that during summer, we don't need to irrigate. I mean, it rains every day, twice a day. So the thing about Jatropha is that, a lot of when I started working with it is the fact that it was an attractive crop because it grows supposedly on marginal lands. We know that, it can grow on marginal lands, but it still needs some input. Otherwise, it will not produce. And track, I mean, the type of soil, we can grow vegetables there and fruit trees. So it's not that bad in that sense, but the plants, they grew really well, and they didn't need much input. We used just regular fertilizer, and again, irrigation when needed. We never spray for anything because we wanted to see exactly if they will have any diseases or pests. We have some minor diseases or pests appearing, but nothing that will compromise the yields. And the other thing, the seeds are toxic. So, and the leaves also have compounds, esters, that are toxic. So I think that in a way, you would see that some insects will start chewing on the leaves, and all of a sudden it would stop. So in that sense, the soils were very good for Jatropha. They grew really well. I just imagine that if we could grow on better soils, they will grow even better.
And I may have missed just a follow-up question. So Boeing, are they still working on this in terms of using biofuels?
No, at one point, Boeing was very interested. And again, when I first was called to do this space experiment, and I was called for a meeting via teleconference, I was with these Boeing executives, people from Honeywell, USDA, NASA, and I talked to them a lot. We had meetings, and they were very interested in developing. The problem is to get Jatropha to yields that are significant for producing sufficient fuel for the aviation, you need first lots of acreage and cultivars that will be highly productive. We're talking about billions of gallons of fuel that are needed, basically daily. So we were not there yet. We were just starting the process. Now, there was still an interest. We're being funded by a private company, and they were very interested in biofuels. I wanted to start, my goal at the time was that if we started small with growers in South Florida, if they could dedicate 5 acres to Jatropha, they could extract the oil and through transesterification produce clean biodiesel, which is an expensive process. And they could use the biodiesel for their tractors, machinery, trucks. And we could expand that to the county for also machineries, school buses, and public transportation, and make it a green county. So that was my vision at the time. But again, oil prices dropped, biofuels disappear from the scenario, and then that's it. Funding disappeared as well.
I know. I just filled my car this morning, and fuel prices are going up again.
I know.
I don't know. Yeah.
Same here. I filled this morning, and I was like, wow, $3.15.
Yeah.
Any other questions?
Hey, Wagner, this is Diego. Great to see you again.
Hey, Diego, how are you? Good to see you.
Just wanted to reiterate, actually, the project that Wagner mentioned about the tissue culture for us has been a great success. We started small, we started simple. Remember, we started with 2 varieties, a few plants, and little by little, right now, it's probably a huge commercial success. As you mentioned, we produce every year 2.5 million plants on a regular basis, which is supplying all the seed needs that we have for our operation. So thank you for all the support and all the work there. I mean, we started from something small, and now it's a full commercial success. And I would say probably the largest lab, for sure in sugarcane, is probably the largest lab in the US, and it's probably one of the biggest labs in the US regarding tissue culture.
I agree. And actually, I thank you because, you guys believe in the project, you funded it, and the results came, and now is a reality for the industry. So I'm glad to see that, to see that transition from basic research to commercial application. That's what we want to see, right?
Hey, Lourdes, you have a question?
I do. I guess you saw me remove my mic. So Wagner, I'm so glad you brought up the Indigo Trial, because it's about a year, and I know we were going to talk about maybe doing a follow-up. What's the status of that? Because that really garnered a lot of attraction.
Yeah.
So I'd like to do that again.
Yeah, again, that's another interesting study that, came from basic concept and just field testing to commercial application. The last that I talk is that Sarah, who's the owner of Stony Creek Colors, she was in conversations with a few growers. I forgot who exactly. And they were trying to get into an agreement to do the field testing in their commercial farmland. So, but after that, I haven't heard anything. Actually, I think it's about time to contact them and do a follow-up and see how that is going at the commercial level. I think I even mentioned to you that it would be nice to have because Lourdes wrote an article about that. And I think now it will be interesting to follow up and write another article to see if that is really working at the commercial level. So yeah, but Lourdes, I don't know exactly what's going on right now. I mean to contact Sarah, but we got so busy and we end up forgetting.
I know. I'll send you an email. Maybe we can follow up together with her.
Sure, be glad to.
Thank you.
I have a question.
Go ahead, Orlando.
Hi, hello. Thank you for the invitation. My name is Orlando Cotto. I'm from USDA Counterpoint, working with before we had it. I have a question about the bioreactor experiments that you developed. Do you have any experience in using beneficial microorganisms to inoculate during the bioreactor? I am talking about biopriming techniques.
Yeah, something like biostimulants.
Exactly.
Yeah, well, not yet, but actually, I'm glad that you mentioned that. I'm in conversation with a company. It's called IQ Biotech. They're from Miami, actually. The owner is from Spain, and they developed these biostimulants. And we're right now talking about doing some evaluations here. I want to do, particularly for acclimatization of plants, coming out of in vitro to ex vitro greenhouse. But also we're talking about using that in vitro in the bioreactors. So that is in our plans. First is the biostimulants that they have. And also I'm interested, they are working with trichoderma. And I would like to see if trichoderma will have a potential for controlling some of our in vitro contamination, but I'm not sure how that will work in vitro. But again, we're designing some studies to address that. I'm very interested in that area. I think it will be interesting.
Yeah, thank you.
You're welcome. As you can see, I do a little bit of everything. It's just I get bored doing the same thing over and over. So I like to branch.
Any other questions?
No, I think it's good to diversify. No, this was really good, Wagner, and also these beautiful pictures. I always go back to your orchid stuff, and hopefully maybe we'll have. I know you offer the small class, the short class in Gainesville.
Yeah.
So maybe we can have you for a short workshop when things are back to normal. I think there's a lot of interest in orchid. Everybody whom I talked to said, how can I keep my orchid alive? It's not like, how can I grow them? How can I keep them alive?
I usually tell people, leave them alone.
Leave them alone, but I know.
The more you do, the worse it is.
I have another question. It's about the Jatropha experiments. The one that you propagated and planted in the field, those one that the oil is toxic.
Well, yeah, that's an interesting thing. The oil for human consumption, yes, from the toxic varieties, yeah. But there is a variety from Mexico that is non-toxic. And I give an example. From the toxic variety, if you eat a cup of seeds, the seeds taste really good. If you eat a cup of seeds, make sure you get a very thick book, because you're going to spend the rest of the day in the bathroom with a terrible diarrhea, and it's really bad. That's why one of the common names is called the Persian nut. And in certain countries, like I think in Jamaica and other countries in Central America, if kids would feel that they are sick of their stomach, the mothers will give them one seed to chew, and that will help kind of cleaning up, right? Now, if you eat more than 2 or 3, 4, 5, you could even die. They're really, really toxic. But this variety from Mexico is non-toxic. And I'm living proof. I ate about 10 seeds and they taste really good, like pistachios. And they use those seeds, they dry them out and they make a flour for tortillas. So we even start looking at, sustainable production of Jatropha for biofuels, but for food. And at that time, I was working a USAID project with Haiti, and we proposed that, and sort of reforest the area with Jatropha trees and use them for biofuel, but also for food. So it was a win-win situation. Of course, because of political reasons, a lot of corruption, it never took off. So the oil of that non-toxic probably is non-toxic, and you can probably use for human consumption.
Yeah, thank you.
Well,
so Wagner, when we talk about using the gravity method or the space and in creating mutations compared to chemical mutation or other mutation, there is some advantage of one over the other, and how they
one big question about plants in space is the effect of cosmic radiation. Even astronauts are exposed to cosmic radiation, but there's not really, I haven't seen a way or any studies that measure that, and if that causes any mutations in plants. So we were not looking at that particular point of view. We're looking at, and maybe I should have explained better, we were looking at gene expression. So what happens is that when those plant cells reach microgravity, it's almost like the cells are sensing an environment of stress, right? They lose the point of reference of gravity, where is up, where is down. And it's like a lot of genes start getting differentially expressed, either upregulated or downregulated. My analogy is that the plant is sensing that something is wrong and is trying to flip switches to find a balance. And when we look at a lot of those genes, there are several genes, but several of them were related to stress response. And some of the changes in gene expression, so you have genes that were overexpressed with stress response genes. And we thought if the plant is enhancing the production of those genes or those proteins for tolerance to stresses, could that have an effect when you bring the plants back to Earth? Would they be more resilient to abiotic stress back on Earth, like drought, flood, or especially in the face of climate change? So that's one aspect that we were interested in looking. But again, we never continue along those lines. But there was a potential there because of the genes that we observed. And then the other aspect was that as the plant reaches that balance in microgravity and it continues developing and growing, we figure, now the plant figure out how to develop and grow in space. So that could be a good application for long-term missions, especially, taking a biofuel plant with multiple applications to Mars that you could, instead of growing potatoes, you have Jatropha too.
Very interesting.
So that's what we were looking at. And I think there is a lot of potential in looking at those things. The problem is that, as you know, getting experiments to space is not easy. There's a line of people trying to do that. I was very lucky because NASA was interested. So, having 5 spaceflight experiments in a period of 4 years was amazing.
Great. Okay, if there's no more question, thank you very much, Wagner, and thank you all for participating in this seminar.